Hello friends, today we will talk about blood proteins as normal and abnormal constituents. And the main objectives of today's deliberation are to know the techniques for separation of plasma proteins, to know the composition of blood proteins, to get knowledge about normal and abnormal proteins in the blood, and the biochemical techniques for the detection of abnormal constituents in the blood. First of all, we will discuss about chemical composition of blood proteins. Plasma, which is approximately 90% water, is a liquid part of the blood and 10% of various substances. Over 100 different substances are dissolved in this straw-colored fluid. Examples of dissolved substances include nutrients, vitamins, salts, respiratory gases, hormones, plasma proteins, and various wastes and products of cell metabolism. Blood proteins, also termed as plasma proteins or serum proteins, are proteins present in blood plasma. Serum albumin accounts for 55% of blood proteins and is a major contributor to maintaining the osmotic pressure of plasma to assist in the transport of lipids and steroid hormones. Globulins make up 38% of blood proteins and transport ions, hormones, and lipids assisting in immune function. Fibrinogen comprises 7% of blood proteins. Conversion of fibrinogen to insoluble fibrin is essential for blood clotting. The remainder of the plasma proteins, approximately 1%, are regulatory proteins such as enzymes, proenzymes, and hormones. All blood proteins are synthesized in liver except for the gamma globulins. Separating serum proteins by electrophoresis is a valuable diagnostic tool as well as a way to monitor clinical progress. Current research regarding blood plasma proteins is centered on performing proteomics analysis of serum or plasma in the search for biomarkers. The normal laboratory value of serum total protein is around 7 grams per deciliter. Plasma proteins are also the most abundant solutes in plasma. Other proteins include enzymes, hormones, growth factors, which are necessary to clot the blood when bleeding occurs. Plasma proteins are not taken up by the cells to be used as food fuels or metabolic nutrients, as are the other solutes such as glucose, fatty acids, and oxygen. The composition of plasma varies continuously as cells remove or add substances to the blood. Assuming a healthy diet, however, the composition of plasma is kept relatively constant by various homeostatic mechanisms of the body. For example, when blood proteins drop to undesirable levels, the liver is stimulated to make more proteins. When the blood starts to become too acidic, that is acidosis, or too basic, called alkalosis, both the respiratory system and the kidneys are called into action to restore it to its normal, slightly alkaline pH ranging from 7.35 to 7.45. Now the types of blood proteins. Albumin. This is the most abundant class of plasma proteins ranging between 2.8 to 4.5 grams per 100 ml with highest electrophoretic mobility. It's soluble in water and is precipitated by fully saturated ammonium sulfate. Albumin is synthesized in liver and consists of a single polypeptide chain of 610 amino acids having a molecular weight of 69,000. It's rich in some essential amino acids such as lysine, leucine, valine, phenylalanine, threonine, arginine, and histidine. The acidic amino acids like aspartic acid and glutamic acid are also concentrated in albumin. The presence of these residues makes the molecule highly charged with positive and negative charge. Besides having a nutritive role, albumin acts as a transport carrier for various biomolecules such as fatty acids, trace elements, and drugs. Another important role of albumin is in the maintenance of osmotic pressure and fluid distribution between blood and tissues. Now the globulins. By electrophoresis, plasma globulins are separated into alpha 1, alpha 2, beta and gamma globulins and are synthesized in the liver, whereas gamma globulins are formed in the cells of reticuloendothelial system. The average normal serum globulin concentration is 2.5 grams per 100 ml or 3.53 grams per 100 ml by electrophoresis. Alpha 1 globulin. This fraction includes several complex proteins containing carbohydrates and lipids. The normal serum level of alpha 1 globulin is 0.42 grams per 100 ml. Alpha 1 lipoprotein helps in the transport of cholesterol. Orosomucite is rich in carbohydrates. It's water soluble, heat stable and has a molecular weight of 44,000. It serves to transport hexosamine complexes to tissues. Lipoproteins are soluble complexes which contain non-covalently bound lipid. These proteins act mainly as transport carrier to different types of lipids in the body. Alpha 2 globulins. This fraction also contains complex proteins such as alpha 2 glycoproteins, plasminogen, prothrombin, heptoglobulin, ceruloplasmin and alpha 2 macroglobin. The normal serum value of this fraction is 0.67 grams per 100 ml. Plasminogen and prothrombin are in the inactive precursors of plasmin and thrombin respectively. Both these proteins play an important role in blood clotting. Heptoglobulins are also glycoproteins having a molecular weight of 85,000. These are synthesized in liver and can bind with any free hemoglobin that may arise in plasma due to lysis of erythrocytes and thus prevent excretion of hemoglobin and iron associated with it. Ceruloplasmin is a glycoprotein synthesized in liver and is an important component of copper metabolism in the body. Nearly 95% of plasma copper is bound to this protein. Beta globulins. This fraction of plasma proteins contain different type of beta lipoproteins, which are very rich in lipid content. It also contains transferrin, which transports non-heme iron in plasma. The normal serum value of beta globulins is 0.91 grams per 100 ml. Transferrin is an iron transport protein. In plasma, it can be saturated even up to 33% with iron. It has a low content of carbohydrate. Gamma globulins. These are also called as immunoglobulins and possess antibody activity. On the basis of their electrophoretic mobility, they are classified as IgG, IgA and IgM. Now the fibrinogen. It is a fibrous protein with a molecular weight of 340,000. It has 6 polypeptide chains which are held together by disulfide linkages. Fibrinogen plays an important role in clotting of blood, where it's converted to fibrin by thrombin. In addition to the above mentioned proteins, the plasma contains a number of enzymes such as acid phosphatase and alkaline phosphatase, which have great diagnostic value. Fibrinogen is globulin of very high molecular weight that can be precipitated easily and can be converted to fibrin, which causes the blood clot formation. Now the functions of blood proteins. First of all, protein nutrition. Plasma proteins act as a source of protein for the tissues whenever the need arises. Second, osmotic pressure and water balance. Plasma proteins exert an osmotic pressure of about 25 mm of Hg and therefore play an important role in maintaining a proper water balance between the tissues and blood. Plasma albumin is mainly responsible for this function due to its low molecular weight and quantitative dominance over other proteins. During the condition of protein loss from the body as occurs in kidney diseases, excessive amount of water moves to the tissues producing edema. Third is buffering action. Plasma proteins help in maintaining the pH of the body by acting as ampholytes. The fourth one is transport of lipids. One of the most important functions of plasma proteins are to transport lipids and lipid-soluble substances in the body. Fatty acids and bilirubin are transported mainly by albumin, whereas cholesterol and phospholipids are carried by the lipoproteins present in the beta globulins. Fifth one is transport of other substances. In addition to lipids, plasma proteins also transport several metals and other substances like alpha-2 globulins, transport copper-bound hemoglobin and thyroxine, and non-heme iron is transported by transferrin present in beta globin fraction. Calcium, magnesium, some drugs and dyes and several cations and anions are transported by plasma albumin. Sixth one is blood coagulation. Prothrombin present in the alpha-2 globulin fraction and fibrinogen participate in the blood clotting process as follows. Prothrombin is converted into thrombin in the presence of thromboplastin and fibrinogen is converted into fibrin in the presence of thrombin. Now the protective function. Immunoglobulins combine with foreign antigens and helps in the removal of these antigens. Enzyme inhibitors remove enzymes by forming complexes with them. For example, alpha-1 antitrypsin combines with elastase trypsin and protects the hydrolytic damage of tissues such as lungs. Some proteins increase during acute phase and protect the body. For example, alpha-1 antitrypsin, alpha-2 macroglobulins. Now the importance of plasma proteins. Measurement of some specific plasma proteins gives useful information for diagnosis and management of some diseases. For example, transferrin receptors, that's ferritin, thyroid binding globulin, sex hormone binding globulin, haptoglobin, albumin, globulins, C-reactive protein, immunoglobulins, and etc. Characteristic changes in amount of certain plasma proteins are seen after surgery or trauma or during infection or tumor growth. Proteins involved are called acute phase proteins. Acute phase protein response leads to greatly increased de novo biosynthesis, that's mainly in liver, of some plasma proteins, along with decrease in the levels of other proteins in plasma. Response is stimulated by release of cytokines, interleukin-1, interleukin-6 and tumor necrosis factor and increased plasma levels. Acute phase protein response is an adaptive response to disease. For example, increases in plasma levels of CRP and complement will contain and eliminate infection. Increased coagulation factors will aid and prevent excess blood loss. Protease inhibitors will prevent the spread of tissue necrosis when damaged cells at the site of injury release lysosomal enzymes. Isolation of plasma proteins. Ultracentrifugation of plasma proteins at 60,000 per minute results in the isolation of blood proteins from the blood. The boundary between the solvent and the protein is visualized by an optical system called Cehlorine system. Electrophoresis. Protein in aqueous solution are charged groups, for example, carboxylic amino groups that can be separated under an electric field using various stabilizing media. Amino groups undergo ionic dissociation at alkaline pH and carboxylic undergo dissociation at acidic pH. Most proteins are negative at pH 8.6. The pH at which positive charges are equal to negative charge is characteristic for a protein and is called isoelectric point. Blood protein associated diseases. The normal range of albumin in blood protein ranges from 3.5 to 5.5 grams per deciliter. Increased level of albumin in blood results in dehydration, shock, and hemoconcentration, and decreased level of albumin level results in symptoms of malnutrition, malabsorption, and nephrosis. In a rare genetic disorder, alpha-1 lipoprotein deficiency results in dangerous disease. If its level is reduced, which causes the accumulation of cholesterol that esters in tissues, resulting in tonsillar enlargement, hepatomegaly, and lymphadenopathy. Alpha-2 macroglobulin make up most of the alpha-2 globulin fraction. In nephrotic syndrome, it's retained in serum and levels are found to increase. Beta globulin deficiency results in inability to transport lipid from intestine or the liver. It also resulted in plasma cholesterol deficiency. It's clinically characterized by intestinal malabsorption. Under progressive ataxia, retinitis pigmentation, and crenation of erythrocytes. The normal range of globulin in blood protein ranges from 2.0 to 3.6 grams per deciliter. An increased level of globulin in the blood plasma results in liver diseases, cirrhosis, malaria, lymphoproliferative disorders. Normal range of fibrinogen in blood plasma ranges from 0.2 to 0.6 grams per deciliter. The fibrinogen level above normal range results in glomerulonephritis and nephrosis, and decreased level from the normal range in the blood plasma results in DIC, placental injury, and meningitis. The deficiency of alpha-1 antitrypsin in blood protein results in obstructive pulmonary disease and liver disease. Deficiency of antithrombin in blood plasma results in thrombosis. Deficiency of immunoglobulins results in severe recurrent or chronic infection. Now the hypoalbuminemia. Hypoalbuminemia is a medical sign in which the level of albumin in the blood is abnormally low. It is a type of hypoproteinemia. Albumin is a major protein in the human body, making up about 55 to 60% of total human plasma protein by mass. Many hormones, drugs, and other molecules are mostly bound to albumin in the bloodstream and must be released before becoming biologically active. Hypoalbuminemia can also be present as part of the nephrotic syndrome, in which protein is lost in the urine due to kidney damage. Low albumin levels can be an indicator of chronic malnutrition or protein losing enteropathy. Hypoalbuminemia may cause generalized edema, that's swelling, via a decrease in oncotic pressure. The serum albumin level is part of a standard panel of liver function tests. Levels below 3.5 grams per deciliter are generally considered low. Now the main reasons for hypoalbuminemia. Decreased synthesis may be due to malnutrition or malabsorption. It may be a feature of advanced liver disease, abnormal distribution or dilution. It may be induced by overhydration. It may be caused by increased capillary permeability as occurs in septicemia. Abnormal excretion or degradation. It may be caused by nephritic syndrome protein-losing enteropathies, burns, hemorrhage, catabolic states. Plasma protein tests. Plasma protein tests are blood tests that detect the amount of proteins in the blood. Such lab work is often ordered as part of a comprehensive metabolic panel during a physical exam. This screening tool can help a doctor to determine overall health. Also called total protein or albumin tests. Plasma protein tests may also be ordered if doctor suspects that you have a certain underlying health conditions such as inflammation or certain autoimmune conditions, which includes rheumatoid arthritis or celiac disease, etc. Now the functions of plasma protein tests. Doctors order plasma protein tests in order to measure the amounts of specific proteins in the blood. Total protein levels may be higher or lower than average in the case of certain health disorders. These include bone marrow disorders, edema, which is a build-up of fluid in the tissues, hepatitis, that's a liver infection, HIV, inflammatory bowel disease, kidney disease, leukemia, liver disease. Results of plasma protein tests. Plasma protein test results often coincide with overall CMP results. Abnormal protein counts can indicate potential health problems. Higher than normal protein levels are associated with bone marrow disorders, infections, inflammation, kidney or liver disease. In addition to albumin levels, proteins test may also detect blood levels of globulin. This is called an AG ratio. According to the American Association for Clinical Chemistry, a normal AG ratio is just above 1, with albumin being higher than globulin. If this ratio is off, it can affect your total protein count. Low AG ratios are associated with too much globulin, which can cause autoimmune diseases. High AG ratios can indicate leukemia or bowel disorders. This brings us to the end of the lecture. Hope you have enjoyed and understood. Thank you and goodbye.